Now we're in chapter 9, and we're going to talk about primary producers and microbes. And then chapter 10 will be animals. On the left is a spirulina, is a cyanobacterium, and it is one that some aquatic organisms feed on fairly heavily and grows in saline systems. And on the right is a diatom that makes glass frustules, uses glass cell walls, and is one of the more beautiful organisms, I think, on the planet. So we'll talk about first viruses, then the archaea, bacteria, protists, fungi, and plants. Viruses, you can argue, are living or not. They certainly can't make it on their own, but other species, no other species can make it completely on its own that we know of. There might be some very deep groundwater species that can live in single species, but it's as a single species, completely isolated. But almost everything depends on other organisms. But they cannot really reproduce themselves. They're basically RNA or DNA, and then a covering of some sort, which can be they can have or not, which are proteinaceous. There's also prions, which are proteins. We don't know a lot about them. They tend to be things that change the conformation of proteins in their hosts and then basically creates more of themselves, and that goes out. And things like mad cow disease are prions. Many aquatic organisms are vectors for human disease viruses, West Nile, Zika, chikungunya. So there's mosquitoes are, a big problem, but there's other organisms that can transmit viruses. This is one thing that has changed since I started, many years ago now. I started in the field of freshwater ecology and marine biology as well. And I have a colleague that was looking at, they were looking at filters, and they were looking at what got caught on filters, and they were looking at the sizes of filters actually and how they worked. But one of the things that they found on these filters when they filtered water from lakes and oceans was what they called virus-like particles. So things that looked like known viruses, yet they were floating around and very common in these waters. And this got them thinking about, what they're doing there and what's their ecological role. And it turns out that if you concentrate these particles and put them in with like an algal culture, you can wipe out a lot of the algal culture because there are some viruses in there that are specific to that alga. This turns out that every organism has its viruses. Viruses have been with us as organisms since the dawn of life. And they've adapted to be parasites of every species out there. So they can be very ecologically important. And they can do things like break cells and cause them to release their nutrients and keep the nutrient cycling, what we'll call the microbial loop, going. And here's the major groups again to bring you back: the bacteria, the archaea, and the eukarya. And the bacteria and the archaea look similar, but they're as different from each other as they are from the eukarya. The archaea, as I just said, they're morphologically similar. They originally were thought to be mainly extremophiles. So they were isolated from hyperthermic environments, halophilic, salt-loving, or anaerobic environments. As we got these ribosomal RNA methods going and started sequencing, it turns out they're everywhere, that you can find them everywhere, and they're quite common. Again, something that we didn't even know there was a separate group of these 50 years ago, and now it's its own super kingdom or domain. They're essential in nutrient cycling, and when we get to nutrient cycling, we'll talk about some things like methanogenesis that have a global effect on the environment, and freshwaters play an important role in that. Tom asked, have we used algal viruses as an ecological control? That's a good question, and there have been attempts at it, particularly for cyanobacterial blooms. And it could explain why we get these big blooms and then they crash. Why would you expect a bloom to be more likely to transmit a virus than a low density algal population in the lake? Easier to spread, right? They're close together. The viruses can't survive very long in the environment. Things will inactivate them. They might get stuck on another particle. So yeah, it's kind of like whether you're walking around campus with a mask on or you go to a football game and sit there without a mask on. So your chances or a political rally, your chances of getting the disease are much higher when you're concentrated. Good. So people have talked about that. Functionally, it's not clear. It's not a common strategy, at least it isn't yet. But it's a possible one that people are looking at. So that's a good question. Bacteria are probably the most important organisms in nutrient cycling on Earth, and we can only culture less than 1% of all species. So that's the other big thing, that being able to sequence ribosomes from the natural environment found out. Even the very first people that did it, the two environments where they were able to take natural ribosomes out of the environment and amplify and sequence them were a hot spring in Yellowstone and the SARS, a marine Sargasso Sea population, and they had not cultured organisms except for maybe one or two of those sequences at all. So there's just a whole new world out there of microbes that nobody knew was there, and based on the fact that we just couldn't bring them into the laboratory and study them. Morphology is relatively limited, except for in the cyanobacteria, which we'll see. Most identification is based on metabolic or chemical characteristics traditionally. So the ability to photosynthesize or to break down protein or to use ammonium or something like that, peroxidases. And the cyanobacteria is a really important group that we'll talk about a lot in this class because they're really central in water quality. So here's the sort of the generals of morphology. You can have a sphere, a rod, a spiral-shaped spirilla. They can be filamentous. You can have a streptobacillus as a chain of cells, of rods. Streptococcus, which you can get that strep throat is an example of one that if you culture it, it looks like the streptococcus. Staphylococcus makes another sheet. Vibrios are curved rods. Spirochetes, Sarcina is a stack of eight cells, a cubical stack of 8 cells. So, we have millions of species of bacteria, yet there's a sort of the range of what you can see under the microscope if you look at them, except the cyanobacteria. And it means that we've got to find another way to do the taxonomy. So we'll get into the cyanobacteria. These are important because they produce oxygen, they're photosynthetic. They were the first primary producers. They oxidized the Earth's atmosphere early in the evolution of the Earth. They made it possible for the multicellular organisms to inhabit the Earth. Because we talked about multicellular organisms are inefficient at diffusion, right? Because they're big and molecular diffusion rates are slow. Once there was oxygen available for respiration, they were able to more efficiently use organic carbon, and if they got a system to bring that oxygen in, they were able to respire and make multicellular organisms. In addition, the oxygen entered the Earth's atmosphere and broke down to ozone in the upper atmosphere. And why is that important, ozone in the upper atmosphere? UV protection, great, Emma. Right, so that's why we're worried about destruction of the ozone. But before the cyanobacteria came up with the evolutionary solution of splitting water for photosynthesis and giving off oxygen as a byproduct and oxygenating the atmosphere, the UV was blasting down. There could be no terrestrial organisms really. The Earth, the terrestrial habitat was not inhabitable or survivable, as well as any shallow water environments as well. They're also important because they have special cells, specialized cells that fix nitrogen with heterocysts. And we'll talk about this when we get to the nitrogen cycle. They're able to create gas vesicles, which are just open vesicles that make them less dense than water, so they float and you get scummy blooms. They produce objectionable odors, tastes, and toxins. So they really have a big effect on water quality. Again, we'll talk about that. And they have unique light harvesting pigments. We already talked about that when we talked about light. Remember, they have specialized pigments that can use green light, so they can live at low light situations. When we got to Potawatomi State Lake number 2, at 6.5 meters, we found a dense population of cyanobacteria. They're living down there where the nutrients are high, coming up from the bottom. The light's low. The green algae above are filtering out the red and the blue, but the green can make it through, and so they can photosynthesize and grow down at that deep area. So this is a start of some of the varieties of morphologies of cyanobacteria that you can see. And I am going to stop, and we will continue our lecture on Friday.